Molecular biology. The term molecular biology was proposed by Warren Beaver. Molecular biology is a modern branch of biology which is formed by the convergent work of physicists, organic chemists, by the work of a physicist, by the work of organic chemists, and also geneticists collectively forms a molecular biology. And the term molecular biology was proposed by Warren Beaver. Warren Beaver. In all prokaryotes, eukaryotes, and most of the viruses that contain DNA as a genetic material. DNA as a genetic material. In some viruses like a TMV virus that is called tobacco mosaic virus, HIV virus, and bacteriophages, RNA acts as a genetic material. RNA involves in many metabolic events in the body. It acts as a messenger RNA and a structural role like rRNA that is called ribosomal RNA that is called ribosome formation. Next, catalytic RNA. So it also participates in the catalytic activity. So catalytic role, RNA acts in the form of ribozymes. Ribozyme is an enzyme. 23S rRNA acts as an enzyme that is called a ribozyme. And lastly, adapter, like a adapter molecule, it acts as a tRNA. Our next topic is nucleic acids. In the year 1869, Frederick Miescher first identified an acidic substance inside the nucleus. So here, if you see, every cell inside there is a nucleus. Robert Brown discovered nucleus. We know already Robert Brown discovered nucleus. Within the nucleus, chromosomes are present. These chromosomes are with deoxyribose nucleic acids. Means in the nucleus, acids are present. This acidic nature of the genetic material first time it is Frederick Miescher. Frederick Miescher coined the term nuclein, but in the year 1953, Watson and Crick identified the double helical structure of DNA. Watson is a zoologist and Crick is a physicist. Watson worked on effects of hard X-rays on bacteriophage multiplication, and Crick worked on the X-ray diffraction studies of proteins. Wilkins and Franklin proved that X-ray diffraction studies of DNA. Pauling worked on the hydrogen bonds present in DNA. E. Chertoff says that purines and pyrimidines are in the ratio of 1:1. So there are 1:1 ratio in the DNA, the purines and pyrimidines. The purines and pyrimidines both are in the 1:1 ratio. This is given by E. Chertoff. Next, coming to the structure of DNA. According to the Watson and Crick, it is a double helix. It has two strands. DNA has two strands, and each strand is a polynucleotide strand. Two strands are anti-parallel. One strand is starting with 3' end and ending with 5' end, and another strand starting with 5' end and ending with 3' end. This is called anti-parallel nature of the DNA molecule. According to the Watson and Crick, DNA is a double helical structure. Each chain is made up of polynucleotides and they are anti-parallel in nature. And Chertoff rule is applicable to the double-stranded DNA molecules only. Each strand of DNA molecule is made up of Nucleotides. Every nucleotide is made up of three components. Number one, nitrogen bases. Number two, sugar. And number three, phosphate. In the nucleotides, nitrogen bases are of two types. One is purines, second is pyrimidines. Purines are double ringed and pyrimidines are single ringed. Adenine and guanine, these two are called purines, whereas cytosine, thymine, and third one in the RNA it is called uracil. So in the DNA it is a thymine. So here A, G, C, T, they are called A, G, C, T. These four are the nitrogen bases present in the DNA molecule. Adenine and guanine are the purines and cytosine and thymine they are called pyrimidines. Next coming to the sugar. The sugar is called pentose sugar. Pentose sugar. So this is the first carbon, second carbon, third, fourth, and this is the fifth carbon. The 5 carbon pentose sugar present in the DNA, and DNA full form means here deoxy. Here DNA means deoxy. Deoxy means without oxygen. So here in the second position, in the second carbon, instead of OH, O is absent, only H is present. So here only H is present in the second carbon, so it is known as the 2-deoxyribose sugar. 2-deoxyribose sugar is present in the DNA. And lastly, phosphorus. So H3PO4. H3PO4 is the phosphate. H3PO4 is the phosphate present in the DNA molecules. So here every chain in the DNA is made up of nucleotides. The number of nucleotides decides the length of the DNA. Every nucleotide is made up of three components. One is nitrogen base, second is sugar, third is phosphate. Nitrogen bases are of two types, purines double ringed and pyrimidines are single ringed one. Adenine and guanine, these two are called purines, and cytosine and thymine are called pyrimidines. The sugar in the DNA is known as a pentose sugar, the 5 carbon atom. At the second position of the carbon, the oxygen is absent, so it is known as 2-deoxyribose sugar. And the phosphate present in the nucleotide is H3PO4.